Hi everyone, my name's Mac, presenting this work on behalf of myself and Eli Muller. Today we'll be talking about diffuse coupling in the brain and whether we can think about diffuse coupling as sort of like a temperature dial for computation and how we can use computational models to actually help us understand how the neurobiology of the brain might give rise to some of its emergent functions. So in my group, we're really interested in this problem that's been staring us in the face in neuroscience for really the entire time, which is that the brain is organized across many different scales. So we could look at the brain through a microscope and we could find individual neurons and characterize, let's say, different pyramidal neuron classes from one another and talk about their different spiking properties. Or we could zoom right out, a little bit like looking at the brain with a telescope, and look at things like EEG or fMRI data and watch the patterns of resting state networks could form and dissolve over time. And the really fascinating thing from our perspective is that these are both signatures of the same system, yet they look so different. So we could be down at the neuron level or up at the whole brain level. And it's really, really hard to figure out how is it that you could create a link between these scales that appear so disparate. And as part of this workshop today, it won't be any surprise that we're going to argue that models are a really great way to do that linking. So in particular, in my group, we're interested in this property of diffuse neural connectivity. So we often think about neurons as these kind of point processes linking one another. The analogy that people often use is the sort of message being passed from one neuron to the other. So the retina passes the message to the lateral nucleus, passes a message to V1, which passes a message to V2, etc. But there's also these really interesting diffuse coupling patterns that are pervasive in the brain. The two that we think a lot about are the ascending arousal system. Here we've used the locus coeruleus as an example for that. This is unmyelinated fibers that project out through the whole brain, quite widespread, releasing a chemical known as noradrenaline or norepinephrine if you're in North America. And the thalamus also has a number of these really diffuse projecting systems. And so we're really interested in how we can understand the role of these. They're not really like message passing. What's the best analogy we can use for these? Now, the reason we are interested in them is that they're both really important for these really broad categories of function, flexible information processing on the one hand, and what we would term complex adaptive dynamics. These are sort of wrapper terms for these really interesting behaviors like flexibility, switching, the ability to parallel process, to conduct multiple tasks at different times, the difference between explore and exploit dynamics. So really big catch-all terms for the sort of complex adaptive dynamics. So the ascending arousal system, I mentioned the locus coeruleus. There's a bunch of different nuclei that comprise the arousal system. Each of them have their own little idiosyncrasies. Just on the left, just a few of these that you might be aware of, things like the dorsal raphe nucleus, which projects serotonin. There's the paraactiductal gray, the tuberomammillary nucleus, which projects histamine. The basal forebrain is another one we're really interested in, which projects acetylcholine, but also GABA and glutamate around the brain. Lateral hypothalamus projecting orexin. So all of these have in common the fact that they are autonomously active nuclei during wake, and they also project out diffusely to the whole brain and release neurochemicals. People have linked the function of these different circuits and relative activity patterns between them in what we would call flip-flop mechanisms. So there's really famous work on this, thinking about these really big, massive state changes that happen when, let's say, something like the ventrolateral preoptic nucleus becomes active. That's a sleep-on nucleus. It usually is active when areas like the locus coeruleus, the dorsal raphe, and the dopaminergic nuclei are all off. And so you can imagine sort of flip and flop back between those controlling sleep-wake cycles. The thalamus is organized kind of differently. It's a sort of subsection, sub-collection of all these different little nuclei. They're all kind of packed in tightly together into this kind of weird little egg-shaped structure that's deep in your brain. And there's lots and lots of different subnuclei within the thalamus. They all have different names that usually define on the projection patterns. So the lateral geniculate nucleus will receive inputs from the retina and project out to areas like the superior colliculus and V1, whereas an area like the pulvinized draining areas of the temporal lobe, the parietal lobe, the occipital cortex, as well as collicular input patterns. So these all have different kinds of names and structures. They're actually embedded within a broader topological circuit as well that innervates the cortex with the basal ganglia and the thalamus on the left, as well as the cortex with the pons, the cerebellum back to the thalamus on the right. They've also got this really interesting inhibitory shell around them called the reticular nucleus. There's another structure called the zona incerta, not shown on this diagram, that inhibit and blanket these predominantly glutamatergic structures in inhibition. But the thing that's really interesting to us is that they have these different projection patterns. Some of the thalamic nuclei project in a quite targeted fashion, often to the granular layers of the cortex. We'd call those a core type, and others project quite diffusely, often to the supragranular and infragranular layers of the cortex. We'll call them a matrix type. Another way to think about the thalamocortical system is whether or not the thalamus is receiving predominantly inputs from the external world. We'd call those a first order nucleus, or whether or not they're embedded within a cortico-thalamocortical loop. So they receive a cortical input, then project back to another area of the cortex, and we call them a higher order nucleus. And there are lots of different ways to kind of carve these up. Irrespective, one way to think about what the thalamus can do is that it can support a whole bunch of different functional primitives. So the thalamus is good at prolonging activity in target areas, maybe by allowing that particular area to become active while inhibiting other areas around it in a process we would call divisive normalization. It can support topological patterns by allowing certain regions to become active and then keeping them separated topologically from other coalitions of neurons that are also active. The way it does that is through these coupling events by allowing communication in different kinds of temporal subdomains. So you can imagine allowing two different nuclei to become active in a particular frequency band, like a gamma frequency band, which might give rise to some of that communication through coherence that we know about through the work of Pascal Fries. And interestingly, the thalamus can actually promote neural variability as well. So by being active, the thalamus can actually support the increased degrees of freedom in a targeted structure in the cortex, which is also a really interesting capacity. So these are all more theoretical ideas about how these systems work. So one of the questions that we think about a lot is how can we test these ideas? How can we essentially take these concepts that we see in the thalamus in the ascending arousal system, particularly these diffuse coupling systems, and how can we think about how they might work at the global scale? What kinds of functions could they support? So for that today, what we're going to do is focus on some of the modeling that we've done using neural field theory. So if you're in person, you'd be able to see Eli present this far more elegantly than I will. But I'll stumble my way through some of this information. If you're interested in any of these details, please reach out, double click on any of these questions you have, and Eli would be happy to answer any questions offline. So the idea with neural field theory is that if we've got this complexity of neurons down at the microscale, we can't really naturally track them all around. In fact, if we try to do so, we kind of run into this massive problem of just way too many variables. So one of the things we'd like to do is make some approximations about how they would work at that broader scale. And so one of the things that we can do, one of the really simplest things we can do, is we can take information about an individual neuron. On the left-hand side, we've got the amount of current that we put in on the x-axis and the spike rate of the neuron on the y. And we can convolve that with a Gaussian function, which is, we can simply just track around the mean of that Gaussian, or the width or the standard deviation of the Gaussian. And in doing so, we're making an assumption that each of the neurons that we're putting into this little population are independent, which is obviously an assumption that may or may not hold true in reality. But the benefit is, if we make that assumption, we can now track a single number around, and we can look at, now we have this population firing rate on the right-hand side, this sigmoid curve, where the amount of input that's coming in, i.e. the membrane potential, can track in a nonlinear way to its firing rate. And so it turns out that we can, this really makes the mathematics of the whole process far easier. So the way we do this mathematically involves activation functions and wave propagators. And so the really cool thing is that the mathematics of this stuff is really, really well understood at a sophisticated level. And so even though we're making some assumptions about independence of a population, we can then rest those assumptions on really, really rich mathematics, which have been well worked out. So we can take a little neuron here. We have a mathematical equation that helps us understand how we integrate the different potentials that all come in. We then have a way of translating those into postsynaptic potentials, and then that gives us essentially a dendritic response function. And then you can imagine tuning different elements in these equations to give you either fast responses or slow responses. That'll be mimicking things like AMPA or NMDA receptors, or GABA A or GABA B receptors in the neurobiology. So in our case, what we're really interested in is taking these, the strength of neural field theory, and then using it to test neurobiological ideas and neurobiological hypotheses. And so what we did in one of the first studies we worked on together was essentially take this existing neural field model, or a neural mass model really, of a corticothalamic circuit. So what we have here is two cortical populations, an excitatory and inhibitory population, and then two thalamic populations, the excitatory and the reticular nucleus. And they're coupled together using principles of neuroanatomy. And these have been well fit to electrophysiological data across different states of arousal. And then what we can do is we can take these and we can wire them together into a little toroidal structure. And then we can sweep a single parameter, this diffuse coupling parameter. Now, this could mimic something like the diffuse thalamus, or it could mimic the diffuse projections of the locus coeruleus or the dorsal raphe. This is really a little more abstract than precise neurobiology. But what we were really interested in doing was figuring out what benefits or costs would you confer if you swept this diffuse coupling parameter between its regimes. So in other words, what does diffuse coupling do? And the intuition that we were working on when I was talking about these diffuse systems to Eli when we first started working together, Eli had the intuition that this diffuse coupling was working a little bit like the way that temperature works on a glass of water. So as you increase the temperature in water, let's say I'm heating up a flask of water with a Bunsen burner, I'm increasing the kinetic energy of the system. But what that ends up having the effect of doing is also increasing the diversity of the different individual states of the water molecules. Because as the kinetic energy increases, there'll be some unlucky molecules, or maybe lucky depending on how you think about it, that end up getting bounced into in a particular temporal window. They'll think it's a little hotter than those that are the unfortunate loners that have not been bounced into in a particular window. And to them, it appears a little bit cooler. So you both get an increase in kinetic energy, but an increase in diversity. And the idea that Eli had was that maybe as these diffuse coupling systems increase, what you're doing is you're increasing the likelihood that you can have more different neurons, or in our case, neural masses, in that right tail. And the right tail of that distribution is now going to have interesting information processing benefits that you can take advantage of. And obviously, if you turn it up too high, the whole thing turns into the second state. All of the water becomes gas. They sort of run away from each other too much. And so this was this interesting intuition that changing diffuse coupling could give rise to this diversity of states, which might confer information processing benefits. So we were able to essentially, so here's the way of looking at this. You've got a subcritical regime down here, which is essentially like being a cool glass of water. As we start to heat it up with this diffuse coupling term, it's like the diversity of the state starts to increase. And if we go just a little bit closer towards that bifurcation point, now a set of the neural masses can essentially leave the enslavement of that lower attractor, raise up to the upper attractor. But importantly, the majority of the population doesn't actually follow along. So you actually retain some stability in that subcritical state. And so we call this quasi-critical because it's the system using criticality to compute, but without forcing the entire system to switch over into a supercritical state. So yeah, the questions are, what do we find in the model? So first of all, Eli essentially computes steady states, and then he sweeps the input to these individual neural masses to find their bifurcation point. And then what he can do is he can track the proportion of these different neural masses that have crossed their bifurcation as he sweeps this diffuse coupling parameter. And unsurprisingly, he finds a really nice sigmoid curve as he does so. We call these three different states a subcritical state, a quasi-critical state, which I was just describing, and then the saturated state. For those of you who have more of a neurobiological bent, and this was really for me when we were working on this together, we can then map these back to the kinds of characteristic circuit diagrams that we would see in the individual neural masses themselves. And so being in the subcritical state is like having a really strong excitatory input from the cortex to the thalamus, but having the reticular nucleus essentially have a high activity pattern than the relay nuclei. So even though you're trying to convey a message through the thalamus, it essentially gets blocked. In the saturated state, that's like the sensory relay nucleus winning out over the reticular nucleus. So now the messages get passed through, and this essentially is a state with very little information processing. All of the individual masses jump up to that higher attractor, and you can't really convey any information. So we can do a number of different analyses here. You can look at the time average activity connectivity patterns, like a functional connectivity pattern, and then plot them embed them in a little network diagram, and you can essentially see that the toroidal structure's really clearly present in the subcritical state. You have this kind of big, heavy mass in the saturated state, but in the quasi-critical state, you can kind of have some of that toroidal structure, but it's got an interesting kind of density to one side. And if you watch the little movies of these, you can sort of see the thing moving around. Eli can go through and do some analyses where he takes different different parts of the population that are either closest to or furthest away from the bifurcation, and he can inject a little unit current, and he can essentially find that, to cut a long story short, this quasi-critical zone is the one in which you see the most bang for your buck, the highest gain in terms of the responsivity of different individual populations to that input. And a nice analogy for this is one that we use quite a lot in the lab, which is this idea of an attractor landscape. So you could think about being in a subcritical state, like being enslaved to the lower attractor. As we turn up the diffuse coupling, we're flattening out the attractor landscape. Now, some neurons can jump into that second attractor, but the vast majority stay in this lower attractor. As we flatten it even further, now more and more populations can jump over into the second attractor. And if we go even further, we flatten it even further. Now we've lost the ability to go back to the first attractor, and we're all stuck in the second attractor. So with all this in mind, a question then arises: can we find evidence for this in typical neuroimaging data? So the answer is yes, and the yes asterisk, because it's still an ongoing process. But I think we've found some really interesting hints in this direction with some experiments that I'll just quickly run you through now. So one experiment that we did early on was led by Brandon Munn, a really talented postdoc in my group. What Brandon did was he took 70 resting state fMRI data from a collaborator, Luca Kochi, up at Queensland Institute for Medical Research. We extracted time series from the basal nucleus of maynard, a cholinergic projection nuclei, and the locus coeruleus, a diffusely projecting nuclei. And then he was able to basically take the bold data from the rest of the parcels in the cerebral cortex, and then estimate something like an attractor landscape flatness. So I won't go through this in great detail here, but the basic intuition is that if we map out all the different ways that the brain state could change, where the brain state is defined by the configuration of bold patterns across our set, so let's say 400 different regions from a parcellation, we can ask how stable was that, how much did it change in terms of its mean square displacement, either soon in time or later in time, and how likely was that? And we can essentially use a Boltzmann distribution to take the probability, invert that, and then have some estimate of an energy that it would be required to get to that state. So the reason I have these little activation energy paths from year 11 chemistry up the top is that the intuition here is very similar. That the idea would be that if you put in a diffuse coupling term, what you should do is make it easier to transition to different states. It should be a flatter attractor landscape in this sense. And if you were to put in a cholinergic tone, you should essentially increase the energy barrier. It should be harder. So Brandon could then go look in the data at the points in time where there were peaks in the locus coeruleus or peaks in the basal nucleus of maynard, and essentially calculate these landscapes. They're hard to look at in 3D. So if we slice them, let's say at the 10th TR, I can then show you that the energy required to move further in space at a particular delay is actually much lower for the moments after the locus coeruleus peaks and much higher after the basal nucleus of maynard peaks. So there's a really different way of looking at neuroimaging data, but it shows you that you can actually find evidence of these kinds of concepts that we're seeing in computational modeling in neuroimaging data. Just to show you that it's not a just a descriptive measure, we've actually got causal evidence now from a really cool study led by Valerio Zerbi in Switzerland. And so what they did here is they were able to optogenetically stimulate the locus coeruleus in a group of rodents that were lightly anesthetized in a variety of different fashions, while also recording whole brain bold imaging data. And essentially we could then recreate our analysis. So when there's a sham trial, i.e. when they turn on the light at a different frequency that doesn't stimulate the locus coeruleus, you really see no effect before and after that light's turned on in the attractor landscape. But when you stimulate the light at particular pulses, you can see a flattening for 3 Hertz, 5 Hertz, and 15 Hertz. It's essentially consistent with this idea. So this is a really nice empirical confirmation of the predictions from the models and the imaging work. We've also been doing some recent work on a really cool data set from the NIH that Katie Chang shared with us, where macaque monkeys had muscimol, a GABAergic agonist, placed into the nucleus basalis of main, the main cholinergic input to the cerebral cortex. And our prediction here is that if you inhibit the NBM, what you should do is you should see a flattening of the attractor landscape. And this works in prep, but it looks like there's really lovely evidence for this. Natasha has a poster at HBM main meeting. If you're interested, look her up, and you can go chat to her about the nitty-gritty of this stuff. So that was for the arousal system. What about for the thalamus? So this is some work we did some years ago, looking at a really interesting task like Sudoku, where you have to essentially find the missing piece in a puzzle that's a particular shape, and only the shape can only be on one line and one row at a time. So it can be easy in the case where you just have to find the missing piece, versus hard, that's the quaternary case, because you have to look over multiple different rows and columns to find the answer. So we've essentially found really interesting low-dimensional patterns in the data, essentially suggesting that the way that we solve these tasks is by recruiting these sort of low-dimensional networks. But interestingly, the thalamus also did something quite similar. So the dotted line here is the way that this first principal component maps out the different tasks across the different levels of complexity. And both the medial and the posterior compartments in the thalamus here are actually doing something very similar. The anterior compartment looks like it doesn't really care so much. So that suggests that the thalamus is actually tracking the low-dimensional dynamics. But interestingly, if you look across subjects, the more that you recruit the thalamus, the less low-dimensional your brain was. In other words, it was both promoting the integration of the network, but also the segregation. And we think that that balance between integration and segregation is really important for providing an optimal information processing domain. So I mentioned before that we really care about these diffusely projecting thalamic nuclei. In particular, we think a lot about these non-specific thalamic cortical nuclei that Ted Jones described as the matrix nuclei. So here these are calbinin enriched, and they project out to the supergranular layers of lots of different cortical regions at once. And so we think they can play a really interesting role. But let me just skip over that one. That's just some empirical evidence that you can find these patterns relating to low-dimensional gradients. The really interesting thing is that this result has actually now been replicated 3 or 4 times in literature. If you anesthetize an animal, in this case a macaque monkey, and then you go in and electrically stimulate an area enriched with these matrix type projections, you can actually wake the animal up from anesthesia. So it turns out that you stimulate, the animal wakes up, and then you see this increased coherence and coupling between these different sort of regions of the brain that would otherwise be essentially uncoupled during anesthesia. And you can look at a before and after stimulus of the coupling between these different bands. So we wanted to know, how can we incorporate this advanced information, these really interesting empirical results into models of the corticothalamic system? And unfortunately, the way that we'd set up our model beforehand really wasn't sophisticated enough to capture this subtlety, because we had just had this one diffuse coupling term. We hadn't separated out thalamic populations. So Eli was provided with the challenge: can you tweak your corticothalamic model so that it still does all the things that we thought were interesting before, had all the same properties that I described to you in that first paper, but still has this subtlety that we can unpick? And so what Eli essentially did was to take that corticothalamic neural mass and then add in, essentially split the thalamic population into 2. One's the core-like projection that goes back up into the cortex, and the other one's this matrix-like connection that spreads out and has this really interesting connection probability that drops off with distance. And then what he could do is he could, in his model, he could also spatially embed it so that he could create a gradient that we saw in that neuroimaging data, where the core nuclei were supporting more sensory motor ends of the gradient, and the matrix nuclei were supporting more associative areas at ends of the gradient. So then he ran a number of simulations, so you can get activity patterns. That gives you a simulated correlation matrix. You can compare that with an empirical correlation matrix and find really nice correspondence between the two. Eli then had to come up with a way of modeling propofol, which was the anesthetic used in the macaque study. And in this case, the way he did that was by prolonging the inhibitory postsynaptic potential of the inhibitory regions in the system. That actually turned out to be a really nice match to the data. So then he essentially had to come up with a way of embedding this within the brain. And the way he did that, as I mentioned before, was having this really nice connection probability that drops off with distance. For those that are interested, this is related really deeply to this really lovely idea of geometric eigenmodes. There's been a number of papers that have come out recently. Pang et al. In Nature was probably the most infamous. And the idea here is that there are these low-dimensional descriptions of the structure of the brain, irrespective of the function, that give rise to energy states that make it more or less likely for you to be able to activate those different modes. But a way to think about those is that they're saying, what if all we could do was tell you something about the local dynamics and how they propagate? How far could we get? And it turns out you can get really far. So I'll leave this for you to read if you're interested, but there's a deep connection here to some really, really fascinating physics that I'm personally still just getting my head around. So then once Eli had his model, he was able to stimulate the thalamus. And so essentially this is the empirical data. So he goes in and stimulates the matrix thalamus, and then he can recreate the same kinds of things that we see in the data. You can get that same alpha peak, the same coherence between these different frequency bands with high matrix stimulation, but not low matrix stimulation. And he can recover some of the same patterns that we saw in the pre versus post stimulus. Probably more interesting to us is that it wasn't just that measure that we could recreate. If we ran a series of different measures from the literature that have been used before to distinguish between anesthetized and awake brains, we could recover all of those qualitatively. Some of them were a little higher in wake than they were in our model with the stimulation, but the same idea is at play here. If you have propofol and low matrix stimulation, you're low on each of these dimensions. And if you get stimulated, or if you look at wake data, you're high on these dimensions. So we think there's still more work to be done to make this model even a better fit to data. But the fact that we can recover such a broad set of these different terms is really exciting to us. The benefit of these models, though, is that Eli can then go in further steps and unpack some of the really interesting things that happens in, say, the wake versus the anesthetized brain. And one of the really cool things is that the waves that propagate in these models do so differently depending on whether or not you're awake or in the propofol state in the model. In short, when you're awake, you can have a lot of different waves propagating in parallel, but when you're in the propofol state, you have much more serialized propagation of states, almost like there's a little sort of train following itself around versus like a big rock that's thrown in a pond and multiple different ripples occur at once. And so we're really, really interested in that idea and trying to understand it better. To map it back to some of the ideas we've talked about today, we can think about that that idea of the wake state and these different simulations in terms of information theory. So I haven't talked about this much today, but the idea is that in the propofolized state, you essentially have high amounts of information storage, but low amounts of information transfer. You're not reducing the uncertainty of another population with knowledge of the other. But then in the wake state, or when we stimulate with the high matrix, you see a switch from storage to transfer, which is consistent with some other work we've done before in the past. To map that back into the attractor landscape framework, the wake state is like, and the high matrix state are like having a flat landscape. You can move to multiple different states, but the low matrix and the propofol state are like having a relatively rugged landscape. You're stuck in the state that you're in. So we think this is all internally consistent with the previous models. So to wrap up, the idea here is that in the lab, we're really interested in the idea of taking both the microscope and the telescope and trying to find ways to combine them. So taking the neurons and the details about how they work and pulling them together with the kinds of higher order terms that we see when we measure the brain. And we think that diffuse terms like the systems like the thalamus and the locus coeruleus and the ascending arousal system are really important for understanding the link between these two scales. So with that, I'll say thank you very much. Please, if you have any questions, reach out to Eli or myself or anyone from the lab. We're more than happy to take questions. And if you see any of us at the meeting, please stop us and say hi. Thank you very much.